A cracking creation. Jennifer Doudna is a professor of chemistry and of molecular and cell biology at the University of California, Berkeley, and co-inventor of CRISPR technology. She is the co-author of A Cracking Creation. It is intrinsic in humans to enhance their lives and environment. The desire to do this comes in many forms such as literature, medicine, the arts. In fact, enhancing evolution in a controlled manner will be no different because humans strive for perfection. I'm now going to speak about the basic building blocks of life, to natural and controlled evolution. This is what a cell, chromosome, gene and DNA look like. Note that the genome size is not a reflection on the complexity of the being or plant. Humans have 46 chromosomes, which can be found in almost every cell. A mother and father contribute 23 chromosomes each to the offspring. Viruses can implant their DNA into a cell. They have perfected the art through billions of years of evolution. In fact, 8% of human genome consists of retroviruses DNA which infected our ancestors. Like humans, bacteria have evolved an immune system which is able to stop the virus's ability to implant their DNA into a cell and hijack the control center. CRISPR-Cas9 is part of an immune system of a bacterium. It is used to cut double helix, which creates 2 or more sequences of DNA. Professor Kiran Masumuru, who is a professor in the Department of Stem Cell and Regenerative Biology at Harvard, created a technique wherein a cell fuses together a cut sequence of DNA with a desired fit. Combining CRISPR-Cas9 with the cell's ability to fuse together a cut sequence of DNA will enable millions of patients to be cured of life-threatening inherited genes. Prior to the invention of CRISPR, gene editing required remarkable science and expertise and sizable financial backing. Now it can be done by amateurs with no experience in gene editing. This is an example of the accelerating pace in biotech. Computer algorithms have incorporated scientific literature to make gene editing even more simple. This has made it possible for scientists to edit any and multiple genes in the genome, which has resulted in highly complex experiments. Some of these experiments have led to editing genes in cows to make them hornless. Prior to this, a crude and barbaric method of dehorning was used involving the physical removal of the horns. Designer pets will be another possibility. Pets designed with specific attributes and temperament are not out of the realms of reality. Once scientists have mastered this art, they will be able to implement it into other beings. Today, the main method to edit genes in specific places like the eyes or brain is through viruses. Some viruses are not neutralized by the immune system. Thus, if the DNA of a virus can be edited by CRISPR, then use the virus as a means of transferring the edited DNA and have it implant its code into the desired cell. Some of these practices seem controversial, like having designer babies, but ultimately historians will view some people's rejection of these practices as absurd. Their views would be akin to Luddites. The pace of progress will march on. Robert Sinsheimer, then professor of biophysics at Caltech, has similar views. He stated the following: "Man is all too clearly imperfect, a flawed creature. Considering his evolution, it's hardly likely that he could otherwise be. We now glimpse another route, the chance to ease the internal strains and heal the internal flaws directly, to carry on consciously to perfect, far beyond a present vision, this remarkable product of 2 billion years of evolution." The renowned Harvard professor of psychology, Steven Pinker, has similar views. He simply asserted, "The primary moral goal of today's bioethics can be summarized in a single sentence: get out of the way." Bill Gates named biotech as one of the 3 career paths he would consider if he were at college today. He also emphasized that biotech is advancing at a phenomenal pace. What other career paths do you think will be important in the future? Let me know in the comments section. If you would like to see content about emerging tech later, don't forget to subscribe, and to keep me motivated, please do not forget to like.